So we'll make a start. Yeah, you too, Lindsay. Yeah, so we got a rowdy bunch today, which is great. Yeah, so just to start off with an acknowledgement of country. So just to acknowledge that we're on Larrakia lands and pay our respects to their elders past, present and emerging. So we're really lucky today to have Associate Professor Ben Sparrow coming today. You can all read so you can see what we're here to talk about. So TERN network. So Ben's the leader of the TERN group in Adelaide after in a previous life working with both the NT government in Alice and South Australian government. So as you'll see throughout this talk, although most of you are aware from looking at the cross-section of the audience, TERN do a lot of really great stuff with open data and all sorts of environmental monitoring. And I'm sure we're going to hear a whole bunch about that today. So that's all from me, Ben. Thanks a lot.
Thank you. And thanks everyone for coming out in your lunch hour. So feel free to get stuck into a sandwich or something while we're having a chat. So I'm going to start off today by just working out how this all works. Oh, there. Right, now, look at that. There's always got to be a technical glitch done there. So to start off, really big picture, go into a bit of the structure of TERN for just perhaps the first 10 minutes. Allude to some of my colleagues who are in the audience that are far more knowledgeable about different parts of TERN, and then get into more detail on the component of TERN that I lead. So TERN's Australia's environmental research infrastructure. And there's a real advantage in dealing with some of these issues in an infrastructure sense. And so what that enables you to do is enables you to develop teams that specialize in aspects of creating the infrastructure. And in the case that I'll talk about shortly in about, field teams and kit and equipment and that kind of stuff, you can do that centrally at a larger scale, with the idea of collecting once, doing that a really good job of that, and then utilising that many times. That then becomes free environmental infrastructure for anyone to use for whatever purpose they want. And so some advantages of that are that someone else takes care of the costly component of data collection. Many of you have written grants and tried to factor in the field collection. It's a pretty expensive component. So you can make sure someone else takes care of that. You can collect at a scale that no individual research group or grant, etc., could afford. So our program's continental, all our capabilities work right across the country. And so that enables science to happen at a different scale. It enables you to sort of scale up your science, if you like. Because of that scale issue and working in all different jurisdictions, etc., there's standards about how we do our business. So when you're using the data, you have really good understanding and the ability to go and find out in great detail how it was collected, because that contextual information is always important for how you use it. It enables us to do some pre-processing, create not only raw data products, but some derived products that are particularly useful to enable you to answer some of the questions you want to answer quicker by not having to go and do all that pre-work to get the data in a state ready for you. The data can become interoperable and well described because we've got a really wide audience. We make the data available, but also let you know as much as we can about it so you can work out if it's fit for purpose. And I've already talked about the scaling up enables new and innovative and different types of analysis. There we are. Hopefully I'm not auto transitioning here or whatever. So I think when we look at environmental monitoring, it's perhaps an oversimplification, but these are the key components we want to understand about ecosystem change. So where and when's change occurring, what in the environment's changing, what's the direction and magnitude of that change, what's causing that change, and how can we manipulate the system to get change in the direction that we want. And so that then I don't know if I'm doing something wrong here or we're just slow to respond to what the story is. There we are. So each of those types of questions has a type of monitoring that it's well suited to. So for example, landscape monitoring is really good. So broad scale monitoring is really good at those where's change occurring and when's that change occurring. Surveillance monitoring is really good at understanding what in the environment's changing, what's the direction and magnitude of that change. And targeted monitoring, which a lot of the time people use the term environmental monitoring when they actually mean targeted environmental monitoring, but that's the really cause and effect, understanding why something's happening out there and how you would change it. And so understanding those types of monitoring is really key to the way that we've kind of structured TERN. I've got to use this, don't I? There we are. Beauty, getting the hang of this. So let me just press mine. So TERN's kind of structured along those lines. So out to the left there of the screen, you've got a central coordination capability based at UQ. So that manages it, communicates, and there's that sort of management functionality there that coordinates everything. Then you have the 3 data collection platforms. And so you have the landscape platform dealing with that continental scale monitoring, dealing with the where and when kind of issues, those sort of spatial and temporal issues. In the middle, you have the surveillance monitoring, dealing again right across the continent, but looking at in detail at what's changing out there and how much is changing. Sorry, the person that created the slide overly abbreviated it, and I think it changes the meaning. So it's how much as well. And then the ecosystem processes, so looking at that cause and effect component. And so we have 3 components of TERN each that specialize in one of those areas, but then work really closely with the other areas. And I've got a slide to show how that collaborates. But then there's a data synthesis and analysis team that grab the data from each of those capabilities and make it available in the one system and give it all that rich contextual information, tie it all together and enable you to easily access it. So the landscapes component deals with those where and when kind of questions. And so generally leverage sort of remote sensing technologies and some modeling technologies, etc. as well, and give us that information right across the continent. So I think the next slide better demonstrates that. So this slide's intended to show the whole of the maps coloured because there's a lot of the products are everywhere, there's continuous data, but then there's some sites where they go into greater detail to make sure that the calibration validation for those products is appropriate. And so the kinds of products that platform deliver are a whole lot of remote sensing products. So there's land cover dynamics, there's vegetation diversity, fire dynamics, which you obviously know a ton about up here. And there's some specialized field data sets, airborne data sets, and a range of continuous environmental products there through that team. The other component is more soil and modelling based. So the team work on the Soil and Landscape Grid of Australia. And what they do is they spectrally scan all of our soils and run a whole lot of traditional chemical and physical analyses on these as well to develop models of all those attributes you can see there. So a huge range of soil and landscape attributes that they make available as a product as well. That gets quite widely used. Before I go any further, Andrew and Stefan know a whole lot more about this. So landscape questions, you probably bump into these people in the tea room. So feel free to have a chat to them or reach out to me, but I'll probably have to work out who the best person for you to chat to is, because it's not the area I deal with every day. the processes team here, and I'm going to handball all questions there through to Lindsay, because obviously Lindsay works on this. So jump in if I screw anything up, Lindsay. The platform looks at monitoring the environment at a really high level of detail, but at a small number of sites. So really intricate measures using tooling, but at only small representative sites. And so they're done with really intensive field stations that have a range of sensors at. I think I've got a nicer diagram of that in the TIC. But at those sites, they're meant to be representative of a particular environment. And so we've got examples of those environments across the country. We have examples of slightly over half of the major vegetation groups in the country represented by ecosystem processor sites. And so there are the locations of the ones that are supported through the network, though there are others that occur as well for a wide variety of reasons. Just learned about a whole lot more occurring in the last hour or so. Right. So what's monitored? You've probably heard Lindsay talk about this already, I presume. Yeah. So I might skip over this, but the flux towers and the instruments to get at, the next slide's probably more important, to get a real understanding of how the atmosphere interacts with the biosphere and how that interacts with the soil surface. And so those fluxes, those movements through those systems, these technologies enable us to monitor and understand what's going on there. Added to them is a super site functionality. So that's some biodiversity kind of information collected at those same plots. So having the biophysical and the biodiversity information collected at the same location becomes really powerful, because some of those trends, etc., you can see you might be able to link to some kind of biodiversity reason for them happening. So depending on what part we're talking about, some of that information is either collected 6 monthly, annually, or every 5 years. And so they're kind of things like complete species inventories of each of these sites. Biomass, stem size, LAI, ground cover, this kind of sort of traditional field component is collected at these sites at least once every 5 years. And then there's acoustic monitors at the sites as well. And then I've gone slightly out of the structure because I'll come back to the component that I lead. But then there's a data services and analytics team, and their job is to provide access to all of TERN's data. And they also enable people to use some other external data sets we've negotiated to deliver alongside of ours. They use the FAIR principles. We'll touch on that in the next slide. Learned my message when I presented to the territory government yesterday and forgot what FAIR meant. So we'll leave that till the next slide so I can read it. And they provide a visualization process too. So the idea is that they'll develop systems so that you not only can get the data, but you can go in there and you can find out what data is there. You can find out if it's the kind of data you want. You can understand how it's collected. You can make informed decisions about if that's the information you need for your project. And then, of course, access it. And we're continually trying to improve that and make that better, easier, more streamlined process. But obviously, technologies change all the time, so that's an ongoing challenge. They also provide programmatic access. So our data is available via API interfaces where that makes sense. So where you have a longer term, bigger need for the data, we can organise so that we can get computer systems to talk to each other without human intervention, so that you can automate some of this processing as well. They also work on some skills development, try and make people aware of the kinds of data we've got and some of the analytical tools. And between them and my team, there's the development of some of the analytical tools too, so that you not only got access to the data, but hopefully we're giving you some tools to make it easier to actually use that. We'll touch on that point more specifically about the program I run as well. with the overall aim, widespread use. We're just trying to enable people to use our data. So FAIR, findable, accessible, interoperable and reusable. So it's a sort of standard in data availability now, and it's certainly our aim. We also make our data compliant to international standards. The person provided me with the slide had those all those standards listed. They didn't mean anything to me, so I just cut them out, but take it as read. They touch on all those international standards where that makes sense. So they're designed to be fit for purpose, but I think that from this point, the key thing is they're fit for purpose for the research community, but also the management community. And so about 1/3 of the use of our data is the research community. The other 2/3 is various land management agencies, etc., all utilising our data for a huge array of purposes, and we'll touch on some of them in a bit as well. And sorry, I'm not used to not having a screen in front of me, but that's all good. So scalable to deal with large volumes of data, etc., as well, and interoperable with international data capability. So there's a move even in just the last few years to internationalize some of this, have international communities working together. And so TERN, along with NEON in the US, the European capability, and South Africa and a few others are really working to try and make all these systems work together so we can scale up to that next level. I talked about this enabling us to scale up to a continental level. Having those systems talk together enable us to scale up to a global level, which can be really powerful. Huge array of ways to access our data. And this is, as I said, an endlessly moving feast. Some of these are historical, some are being really actively developed and improved. But a great starting place is the TERN Data Discovery Portal, because it's where you can kind of find out about all of our data and it'll send you in the right direction to find it. Some of the others I will talk about later. So we'll just leave it at that. So this one, if that's a great starting point for you. And the reason it's a starting point and not an all-encompassing system, of course, is we've got 3 very fundamentally different types of data. We have really huge volume of raster data, we've got traditional plot-based data, then we have the really not spatially complex, but temporally really complex data. And those 3 data sets need to be delivered in slightly different ways with slightly different standards for each of those communities. And so one place to find them that then sends you to the most appropriate place where those kinds of data are stored. So back to surveillance, and you'll excuse me going on a less than intuitive way of organising all these things, but surveillance is the team I lead based out of Adelaide, and so really keen to chat to you about, a bit more about what our component of TERN is, and because I can actually get into real depth and let you know what's going on there, help develop components of this. So with that middle component, we obviously interact with our colleagues in both those other platforms quite a lot, but looking at those, what's changing, what's the magnitude of change, what's the direction of change. And so that's across a national network of plots and transects, and indeed sort of the transects are a interim stage in that they're the same plots, but their location and the rationale for why they're there is for a particular purpose, and then often there's other things done at them as well. So we aim to provide nationally consistent data and samples, and I think samples was one of the novel things that's not particularly common in these kinds of projects at a larger scale than was previously possible. And so to help scientists and land managers detect change, get to that change. Right, so I'm naughty and didn't update this slide. So it's a couple of years out of date, there's a whole lot more dots on there now. But the idea being that continental, there's data from this network, all states and territories the only terrestrial, arguably, it's probably not terrestrial, the mangroves are the only vegetation community we don't have a number of plots established in. And so getting to all the diversity that's out there. And you'll see in a few of these, we have their orientation is designed at areas we're expecting more rapid environmental change. So these plots are located there because we're expecting change. We want to be able to monitor them more frequently and understand that change. And often there's extra things done at those plots as well. So that's the incorporation of our transect network. So some of the key components of our surveillance is, for starters, surveying the right places. And so that's done by a range of analyses that we've conducted. And so I think the slides reorientate itself. So I hope we've got a little bit clipped off. I can make it available afterwards if that's necessary. So the first of all, we looked at how we'd historically put out plots and if that made sense and how that related to an ideal scenario, and it achieved really quite well. But then we worked out new and better ways using modelling of which parts of environmental space aren't as well represented by a plot network. And then for the last 3 or 4 years have been gap filling, using this analysis to work out environments that aren't well represented and focusing on that, rerunning the analysis, which is the next biggest gap, and gap filling in that sense. But then also working out which plots are most important to revisit more frequently. And it's perhaps no surprise that the understanding of those transects, that some of those areas come out as those areas where we're expecting that change happening. So there's a subset of plots we'd like to monitor more frequently. We've now got an understanding of which, when we're starting to have an understanding of which communities change more rapidly. So we analyze our data and work out which plots within the network, because of the kinds of systems they are, that are inherently more variable, and so we should be monitoring them more frequently. And then the last one, the power to detect change. We're looking at a small number of variables at this point in time and then working out how powerful it is to detect and find amounts of change. And that's ongoing. This is all based on collaboration. So this is not something we've sort of cooked up in some dark lab somewhere. The process started with a couple of years of collaboration with state and federal agencies, academics from across the country. This is, I use this photo because it was one of the earliest stages we did that, but many of you have been involved in these over the years. This has been ongoing, where we started out and the methods and how the programs runs quite a consensus view. And then more recently, we have checkpoints where we have gatherings again of key people that help us with the program. I think you might be in there somewhere, Alan, maybe. And touch base on where we're up to, where we'd like to go, key things we need to focus on, etc. So it's kind of, there's a really strong social component to it as well. One of the key things we do is we collect objective standardized methods for what we do. And so that started out with the rangelands protocol that's on the right there. That did a really good job of detailing how you should collect this information in the field. We didn't realize at the time, but it was a bit remiss of us that the rationale wasn't included in that. So the subsequent paper got to all that rationale and sort of should be used with the methods. And then has subsequently that has been quite well accepted and been uptaken in a range of other things. And we'll get to them in a tick, but it's expanded way beyond its initial intent there. I like to use this slide to get at why we have to actually measure. And so you can see on the left of the screen there, the difference between the, so obvious, sorry, it's pretty obvious, but the transition of Coke bottles through the years. And so you can see determining the difference between those two, and have a go at doing that verbally. You know, I think we probably don't need to measure that. We probably just describe how those two are different, because there's a large change. You get further down the process and we're dealing with much more subtle change between them. And so actually articulating the change between those two Coke bottles is much more difficult. So we probably want to actually measure to quantify that change. So for subtle change particularly, we need to measure. The core of what we do is a hectare plot. So 100 by 100 metre plots. The corner, they're oriented to the grid, so remote sensing people, that makes your life a little bit easier, I think. So north, south, east, west orientation, wherever possible. There's always, as with anything ecological, there's always exceptions to the rule. But wherever possible, we'll do that in a homogeneous vegetation community. The greenish dots there are star pickets that we put in and leave out there. And we survey this in with differential GPS in a purpose-written app. And so we start at the southwest corner, tell the app that this is where we want to start, and it tells us to about half-metre accuracy where the rest of those points are. So you wander around and put the next point in, the next point in. Makes site establishment so much easier. These are just pegs that we use because these are transect lines. So as we do our work, we run out tape measures along there, and we'll get to what we actually do with that shortly. On the right there is a voucher sample. So every species at every plot is vouchered. We take a sample, and I was laughing about this with the team yesterday. We put the barcode on the sample in the field, and essentially from the point of collection create a chain of custody. And while that seems like a great idea, the rationale for that was because I'm just a terrible botanist and needed to work out a system where I could work out what things were later. And so, but we've kept that going because it makes a ton of sense. And so those barcodes are scanned into our app in the field, and that ties that sample forever to the rest of our data collection. Those samples then go off to a herbarium and are ID'd by an identification taxonomist. And if those samples are of particular use to that herbarium, they can access them into their collection, as long as we agree that they keep their original identification as well, and we know about when they're used. If they're not interesting, because we don't have some of the flexibility that a herbarium would to collect at the perfect time for some of those diagnostic features, so we will collect whenever we're out there. If they're not interesting to them, they send them back to us and we keep them forever. And that has a range of reasons. The samples in themselves are useful for a whole range of uses. We've had some taxonomic use already of those, so people can apply and loan the samples. But one of the really interesting things, I think, is that kind of future-proofs your data. So I think of the traditional methods of ecological data collection that we're probably all trained on, where you go out and you work out with a key and you just call it something in the field. That data kind of has a half-life. At some point, it's half as useful as when it was collected because taxonomy changes, etc. So taxonomists, as they do, change their mind or have a rationale for redescribing things. We can go back to the actual samples, re-identify them, keep our data up with the current taxonomy. We also take genetic plant tissue samples. We're trying to run with plant tissue samples as a more generic name because they can be used way beyond genetic uses. There's more recently they've had a heap of stable isotope analyses on them as well. So every species, and from the individual we've taken the voucher sample, we take the first sample for this kind of purpose. They get in a synthetic tea bag, so it doesn't have any genetic component, dried and stored, and then are available for people to use. But if the species is dominant at a plot, and we describe dominant as 5% cover, 2% FBC, if it's dominant at a plot, we'll take another 4 replicates from different individuals across the plot to enable researchers to, they're more likely to be used, so it gives us some redundancy for the samples for all the analyses, but also enables researchers to get at some of that population genetics kind of things as well. We collect point intercept data. As I alluded to on the diagram, we have 100 tapes in that sort of crosswalk pattern, and we go along there, and at 1,010 points along there, we note what substrates there, every species through a vertical projection of that point, height, growth form, species, all that kind of information. And so sometimes that's easy on the right, sometimes that gets a whole lot more complex. We have a laser pointer on the painter's pole, so the point on the ground's a dot, so much easier than trying to interpret. And then we have a densitometer to look up into the canopy. And densitometer, sort of, if you haven't come across one, conceptually it's like a telescopic rifle scope that someone's put a 90-degree bend in. And so it enables you to look forward and see 8 crosshairs up through the canopy to see what you're intersecting. It's important to note that all of the points we collect too, when we're looking up through, as we're looking up through canopy, when we're within a plant canopy, so we look up and we've determined we're in a canopy of a plant, but we're seeing sky. The intercept isn't with vegetation, but it's clearly within a canopy. We record that as an in-canopy sky hit. And so what that allows us to do is instantly have our data available as opaque canopy cover or foliage projective cover, depending on which is more useful, which you'd like to use. If it's opaque canopy cover, you would include that as a hit for the tree. If it's foliage projective cover, you leave it out. So you can see examples here, you get the patterning. This is of a plot. There's an example of substrate, growth forms. You can see the different growth forms of the plot. Given that it's mostly shrubs, it's not the actual side of the picture. But then you can get down to the species level, really accurate measures of cover for the species in the plot. We also use the basal wedge to collect 9 at 9. This is sort of mimics our plot. At 9 locations across our plot, we take basal wedge sweep to give us an approximation of basal area and the plot level. And we also get the team to interpret the strata, so which are the most dominant species in each of those strata, because that's the one part of a proper NVIS description that we can't synthesize out of our actual data. We collect LAI at some of our plots, and we also collect soil metagenomic samples. So you can see here we skim off the top 3 centimetres of soil at 9 locations across every plot. That goes in a calico bag. The calico bag goes in a heavy duty plastic bag with silica there. And those samples get stored for soil genetic kind of analyses. And we'll touch on one of them a bit later. I'm really surprised at the use those samples I have. They're one of our highest demand samples, actually. We take a whole range of traditional soil measures as well. So one of the things I'll talk about a little later is cross-disciplinary. So we create a pit in the southwest corner, and then we take another 9 little sub-pits across our plot. So we do colour, we do texture, we send my 4-year-old at the time down the bottom of the pit to take a sample. So traditional pit, you can see the horizon there. Mitch were to get his head out of the way. The pit, or a range of other measures there. So all those kind of traditional soil measures you generally want. And then key at the pit, we take 3 bulk density measures as well. For those who aren't soil scientists, bulk density measures is we're using this gadget to take a chunk of soil. I hope there's no pedologist here because I get in trouble with my terminology. A really accurate sample within this ring of soil without destroying its structure. And so you then dry that and you have a really accurate calibration between volume and weight and volume. And so anything else we do with our soil destroys its structure. By having a bulk density measure, you can convert what you find out based on weight back to a volumetric measure. So it's really key to data use. All this led to the rangelands method, but then subsequently in 2015, we got some funding for the government to create some more methods. And so the concept is we have a suite of manuals and you go to them and pick the one that's of use. You don't need to for a lot of things. You don't need to develop new methods. There's some out there you can use. Spend your time actually doing research and just take a method and where possible the data, etc. as well. But more recently, the federal government are really excited about how the program works, how those standard methods have been uptaken, and how that enables the standardisation of our data right across the continent, and have funded us to develop a whole range of standardised methods for them. So this is a project that my team in Adelaide are working on that sits next to our TERN work. And so the idea of that being that for a range of federally funded projects, you'd be strongly encouraged to use our methods. When you use our methods, there's tools and data management systems. We can easily feed that into the government. And then they know what their data is going to look like. They can prepare analyses for them, and ultimately meaning they can make better and more efficient decisions. Because currently, often data is submitted as scanned PDFs or whatever, and it's impossible to understand how it was collected, how it compares to something in another state or whatever. And so that's then progressed and led to us developing methods for a whole range of pest fauna species as well. So like those other methods, run through what they are in half a tick, but we have methods for monitoring the abundance of pest fauna. They focus on modules and protocols. So a module being a component you want to measure, vegetation cover, for example. Often a module will have several methods or protocols within there. So our cover has a really detailed, less detailed, significantly less detailed method. So there's different levels depending on what information you need. But at their core is a step-by-step instruction, so that we try to get consistency in the data by making sure everyone collects it in exactly the same way, and really clearly articulating that is key to that. These are the products, the protocols my team are developing. These are really quite mature. So they're at sort of software sense, they're beta stage. We're actually happy for them to start to be seen by people and provide feedback, etc. We're running through a process because I really don't want protocols to become feral. So if you want access to them, I get my comms manager to provide you access to them, and we just track who's got them, because they're not a finalized product, but we'd like to hear thoughts, etc. on them. If you have any problems with them, we'd love the feedback. We can update them. And then my comms manager knows who's got them, so we can send them out updates. This suite they're really busily working on at the moment. So there's sort of alpha versions of them. They're taking shape. We know what they look like. They're going through internal review with the team, etc. And then, the other point on those though is we're not trying to reinvent the wheel. So where there's something that already exists that makes sense, we'll adopt it and encourage its use, or we'll take where there's many things that exist, and a lot of you will be aware of each of the state agencies doing the same thing, but in a very subtly different way, trying to amalgamate them and get consensus across them wherever possible as well. And so using existing things where that's possible. And then working on these species. So these are only just beginning to really take shape. The team have had their first workshop. They're getting ideas of what the methods need to look like and starting to put that together at the moment. What we're aiming for with these is the sweet spot. So there's scientifically perfect, or there's super rapid, and we kind of don't want either. We want something that gives us enough information to make the kinds of decisions we need, but also is cheap enough to roll out and use at lots of locations as well. And that's always a challenge because people have a preference one way or the other often, and so it's an endless compromise. What's going on here? I think I've thrown everything out by being over this side of the room. Evidently, that's not normally done. So the project timeline, we did some scoping government came to us with the concept, and they wanted to know how much it cost and what it had to involve. That happened in 2020. Started getting some traction in 2021, working out what those methods, etc., should be. Get into writing the first set. This year, they've really refined that first set and developed the second set. Getting into field testing, trialing these things out in anger and all the different environments, etc. And then moving into next year, sorting out the vertebrate pests and finalising field testing in apps, etc. So not only are we developing field methods, they come with an app that enables data collection. So my team upstairs take their protocols downstairs and give them to the tech team, who then write the apps, progressive web apps, to enable the data collection. And those teams work really quite closely together. So you have tech people telling what's possible, you have my field team telling what they desire. Most of the time they compromise. Some of the time I find my tech team doing things that I go, oh, just tell the ecologists it can't be done like that. they try to please. But then those apps will enable rapid, easy collection, but also a data system that sits on the cloud to collect that information and provide it to the government, but also back to the people who have collected it as well. And we're just working out the shape that'll take now. Right, streamlined data management. So conceptually, this is what the system looks like. It's way, way, way more complex than this when you scratch the surface, because of all the technology involved, but this is sort of how it works, if you like. So data collection on an app in the field, and we use all the technologies there. So you can use the camera to scan the barcodes, you can use the camera to take samples in the field, you can tie into a GPS, you can Bluetooth into a more accurate GPS, etc. So use the device to collect the information. It gets stored on here locally. So of course sorry, I'm in the way. Of course we designed this to work offline. It doesn't need connectivity. When it's got connectivity, it'll send data to our cloud data store, where we then have a suite of tools that the team enable management of that data. Interestingly though, of course, it doesn't need nearly as much management as you'd normally need, because you've controlled its collection in the first place so well. But this is the kind of functionality we'd upload herbarium IDs and that kind of thing, and then send it off to the world. And so there's a range of tools to enable that. I've talked about the TERN data delivery portal, and then I think we touched on AusPlots R. So my team based in Adelaide developed an R package that has some mapping functionality. You find out where all our plots are, but also enables you to do some basic visualization on them, some basic analysis on the plots, and also get to the samples as well. So the data from the samples is in there as well. So you can use this to narrow down and find out which samples you want to put an application into us to borrow. There's a process around that too. I'll touch on that in a tick. Most stable version on CRAN, most up-to-date version on TERN's GitHub page. But the concept now of being providing derived products, tooling, etc., and putting data and tools in your hands to get into your work at the same time. EcoPlots is where plot databases are centralized. So it's worth noting that AusPlots are updates every night. My team adding new determinations today, you can see them tomorrow. This system, we enable the data services and analytics team to take our data in. And what's really special, I guess, about this system is it really records and maps all the meaning and methods and that for everything that goes in there. And it combines that with all state government agency data sets, etc. So you can have disparate data sets that you can understand what's going on and see them all in the one system. This is only in beta. We're only just sort of getting this happening now. Encourage you to have a look by all means. Again, don't just sit there and get frustrated. They reach out and we can try and fix it. If there's something you find that's not awesome, let us know and we'd love to get onto it. I speak for Guru and his team. I don't have to get on with it, but the data team who are experts, so they'll get onto it, of course. We integrate with our colleagues. And so whilst my triangle diagram at the start, etc., makes it seem like they're really discrete, they're not. There's things that each of those capabilities does with other ones. So for example, we're just working up a temporal biodiversity program at the moment where we'll work out how to sample a smaller number of plots much more frequently, because there's a gap in our understanding of biodiversity in a temporal sense within the network. We're doing spatially pretty good, but 5 to 10 revisits isn't cool. So for some aspects, we're working out how we'll achieve that. Landscapes and processes work together on some sort of real-time carbon and water products. Really excited, and we'll touch on in a bit, UAV program that we're working with landscapes on to get happening. And then there's products that bring the whole 3 components together as well. As I alluded to, one of the key things of the program is a storage facility with over 100,000 environmental samples. These are a time machine almost. There's an archive going back in time of samples and vegetation, etc., from when we were there. And so those plant voucher samples, the leaf tissue samples, but then the soil samples as well. So traditional soil samples for physical and chemical analysis, and then also the soil biological samples as well. And on the next slide, you can see all those samples are going through all the kinds of soil information we can give as well. I lied because it's actually the next slide. You can see a new purpose-built storage facility that we've just got up and going to store all these samples. I need a better photograph than that because that's probably the most boring photo of the whole lot. You turn around, look the other way, there's a great big 30 by 6 meter cool room that stores all our genetic samples, etc., with some functionality in there to help manage them and that kind of thing. Butte, next slide. Tooling to provide tooling helps with standardization. So excuse the formatting has been screwed up in translation here, but by collecting straight into an app, we control so much of the quality. We don't have to work on transcription. We save so much money. We speed up collection. We improve accuracy. We make people collect data in the ways it was intended. And we enable it to then automate the flow through of the data through the delivery systems. We work on better engagement, so telling people what we do, and it's indeed one of the reasons we're here, but all the great work in the field doesn't mean anything if people don't know about it. So we need to tell people what we're doing and why we're doing. I always most prefer to do that in the field. I'm much more comfortable there than here. And telling really disparate groups about what we do, I find really rewarding. So this is a land care group, I think, that I was explaining, and they didn't understand the concept of basal area. So I was explaining to them that conceptually, if you took a chainsaw to all the vegetation at DBH, it's how much of the stems, if you measured their area, that was basal area. And, oh, chainsaws, ripper. No, no. But they got the concept. But then also, where we're out working in their local community groups that want to see what we're doing, it's not a core part of the program, but enabling them to see what we do is always good, and I love sharing that knowledge. So here's the school from Jabiru came out while we're collecting plots in their vicinity, and where this soil scientist Rick at the time explaining what was happening. The other thing we often do is work with a variety of groups to collect this data. So bush heritage is one we've got a big heap of work with. This is Queensland Parks, etc., that we help them establish plots, and then they'll take on their ongoing management. So they'll collect some information in the interim time to give us some more of that temporal depth, but also then interacting with researchers, making them aware of what we've got, how it can be used, all that kind of thing. We aim to educate and inspire. So we want to enable people to collect the kinds of data we collect as well. So that's not, the intent is never for that just to be my team collecting this data, so that we create those really good methods. but not just throw manuals at people. We also give them field training about how to do that. And you think differently, you all know, you think very differently out there when you're waist deep in a pit to how you do in here in the lecture theatre. So we're really keen to train people in both those environments, because both those environments make them think differently. And so we get out there, we show them how exactly annoying it is to take a bulk density, and how every time you try and pry it out, it crumbles and you've got to have another go, and give them tips and tricks to try and minimize that happening, and get them some of those real practical components and tips and tricks and shortcuts, etc., that you get from years of experience. We run training to do that. There's the sort of more formal presentation as well. And we try and find a location where we can do both of those reasonably close. But also when we get the opportunity, get kids involved in what we're doing, because at some point they're going to have to take over, and we'll be sitting on some sunny beach somewhere or whatever, and they'll be out stomping all over and doing our point intercepting, etc., and appreciating what's out there. We embrace new technologies, and so there's an aspect of consistency in what we do. It's absolutely critical to monitor and make sure you're collecting the same thing. But then there's new tools that can sometimes make that easier or better. So for example, right from the very beginning of the program, we collected photo points in a different novel way. So we set up a photo point in the middle of our plot, but then take complete panoramas using a good quality digital SLR at 3 locations. And then that enables you to do a 3D reconstruction of the plot from those photos. This was done 8 years ago. You can imagine, with just the explosion of machine learning, how these processes have improved just in the last decade. And so I've got meetings booked to talk to AIML, the Institute of Machine Learning in Adelaide, to see if they can update that and make it better. I don't have a slide on here, but we actually got to the stage of research grade. We never got it to operation, unfortunately, research grade, but deriving actual basal area of stems from this kind of technology. And then the other one I'm quite excited about, because we're just getting this some momentum at the moment, but we're developing a UAV program for TERN as well. And so why I'm excited about this is there's 2 reasons I'm chuffed to get this going. The first is, and I was a little skeptical, I challenged the team to make it happen, and they met my challenge within 2 days in the field of collecting each of these plots within an hour. Stop the car, an hour later I want to be able to hop back in my car and go, and they did it within 2 days. So rapid collection means I can get to so many more plots. If I can get to so many more plots, it helps me fix my temporal problem, because I can get less detailed information, but I can get it more rapidly. So it becomes part of the mix where I might be able to only get the really rich compositional data once a decade, but perhaps I can get fractional cover at great detail from a drone every 3 years. Helps me get into that finer level of change. The other reason I'm really excited, and I've probably got examples, so this is the kind of imagery you pull off. It doesn't do it justice seeing it here. You'd have to actually put your hands on the data. Range of multispectral products. We're just starting product development at the moment of all the kinds of things we do. But one of the things I think is relatively easy to do in the field is to get a composition. We can work out what's there. To get to its structure and really understand structure is really tough. And so we now have access to some pretty amazing technology to measure structure using LiDAR on a drone that will help complement our composition, our field studies, with much more detailed information than my field team could get with a DVH tape. And so those 2 components, I think, really mean that UAV capability will play a major role in what we do in the future. So the intent is we kit a team up. My field team will go and stomp all over the place and do their field collection. Everyone will knock off for lunch. I'll be forcing them to have a lunch break. One of the ecologists that's trained to fly the drone will put the drone up while no one's in the plot for half an hour. Come back down and they finish the plot. But also I'll have a drone team, based a team of 2 in a vehicle and some kit, to go around and rapidly remeasure these plots. So to get to a whole lot more plots in a year than a field team could. And the last bit, I know we've been going for a while, but very last bit is some big scale analyses. And so by dealing with this as a multidisciplinary thing, we get to willpower. And then an analogy I love to use in these presentations is you need a multidisciplinary team, because if you're about to have a hip replacement, which of these people don't you want to be involved? Well, my answer is, I want them all involved, because they're all absolutely critical. I can't imagine that without their anesthesiologist, but also the administrator is absolutely critical, because I went in for a hip replacement. I didn't want my appendix out. So when I wake up, I hope I've still got my appendix and my hip's been fixed. So the multidisciplinary nature, having a really wide set of skills playing into one problem, gives real power and allows some really interesting analyses. Some of those discoveries that the program's found, with 3 slides to go, we've refound an extinct grass, so that was quite exciting. Far less exciting was that we found a weed that hadn't been discovered, or hadn't been discovered for a long time. No, this one was refound. This is a completely new weed. Not great, but it's also great to have found it rather than it still be out there and we not know about it. This study was really interesting. That I had some researchers from New York, of all places, contact me, and they found out about our program and our soil metagenomic samples. And because those metagenomic samples were located in places where we had great soil information and really in-depth ecological information, they ran an analysis on the genetics of secondary metabolites in the soil. And so these are things that are most likely to lead to drug discovery. Interestingly, as a result of their study, I appreciate that now 80% of our antibiotics come from soil bacteria. And so they ran a study. The basic premise was, if you want to find drugs from compounds in soils, how do you sample to maximize your chance of doing that? And it turns out, a massively oversimplification of a paper is that you maximize latitudinal diversity, and then once you do that, you maximize the pH range of the samples you're taking. That will maximize your chance of finding compounds that are interesting for drug discovery. The other was our data fed into an international study that looked at forests in drylands. So historically, all our carbon accounting has gone, drylands, there's only grass out there, we'll just run equations as if there's grass. But if you scratch the surface and actually have a look, and I don't know if there's anyone from Alice online, but it's not all just grasslands out there. And so this program looked at that globally, but our data fed into it. And what was really interesting was our data was the only data globally that could accurately assess the method that was used in here, because we had such great detailed data. And so it enabled us to find out that globally, there's 9% more forest than they'd previously thought. And so that's it from me. Appreciate that was a long presentation, and thanks everyone for their time. And my contact details are there if you need to get in contact with me.
Excellent. Let's give Ben a thanks. So thanks, Ben. You answered my question. I was going to say, given that we've gone to time and to manage everybody's calendars, because I'm sure lots of people have got places to be, what the best way to contact you was, but there you go. So I guess the way we'll do this is, if you need to run, don't be shy, you can get up and leave. But if you have any questions, presumably, Ben, you don't mind sticking around for a few minutes answering any questions? Of course, yeah. All right, well, I can run around with the mic for a little bit, but then I'm going to have to run. Does anyone have a question for Ben? Oh, that's easy. Andrew,
hi, I've got a couple of questions about data. Sure. You collect a lot of plant specimens, yep, and you're collecting genetic material, which is soft tissue. Are you collecting seeds and other propagatable material?
So no, there's not specific collections of those, but if at all possible, you collect them for diagnostic reasons. So they are included with the voucher samples where they are there. But I alluded to the fact that often our collection timing's not ideal for that kind of thing. So we get what we can when we're out there.
Yep. And with the soil sampling, when you're remeasuring, how much of that are you redoing, if any of it?
Yeah, so we're doing a subset of it as a revisit. we still haven't nailed the perfect frequency for that yet.
So anyone online, you can type something, and anyone else in the room? Easy, all the questions might have headed out before. But yes, anyway, well, then I'll just take the opportunity just to thank you again, Ben. That was great. Can we just give Ben one more round of applause?